Hi everyone, my name is Dr. Ali Huang and I'm a scientist at Mini PCR Bio, a biotech company based in Boston. Thanks for tuning into my talk today, where I will be introducing you all to cell-free protein synthesis technology and how it could be used on the ISS and other spacecraft. Since not everyone listening comes from a biology background, I will first go over what proteins are, followed by introducing cell-free protein synthesis, describe some really interesting applications of this technology here on Earth and how that could be applied to space, and finally give you a sneak peek of our plans to launch. Before we dive into the details of the science, let's first take a step back and think about some of the goals and challenges of traveling and living in space. There are two big challenges in living on the ISS that biology plays a huge role in. The first is dealing with the impact of microgravity. From studying astronauts on the ISS and the ones that have returned, we know that microgravity has a significant impact on biology and how our bodies work, all the way down to a molecular and cellular level. We need to understand more about how microgravity impacts biology so we can better help our astronauts adapt for living on the ISS or even long-term missions to Mars and beyond in the future. The second challenge is the limited resources, such as food, water, or medicines. For now, we're able to send up the necessary supplies up to the ISS, but eventually we would want to be more self-sufficient up there, both again on the ISS and eventually on more long-range missions. Many of these resources, think plants, animals, medicine, are biological. So just like how we need to understand how microgravity impacts our own body's biology, we need to also understand how it impacts how we might try and grow or sustain these resources ourselves in space. Proteins are an essential part of all biology. So first, I'm going to give a quick background on what proteins are and why they're important. Proteins are intricate molecules that your body makes to provide structure to cells and carry out critical functions to keep you alive and healthy. Examples include antibodies, which can detect and neutralize pathogens like the coronavirus here, enzymes that carry out biochemical reactions such as breaking down and digesting your food, or transport proteins that move materials around in your body, like hemoglobin carrying oxygen through your bloodstream. The proteins made in our bodies are important for keeping us alive, but sometimes scientists want to make proteins outside of the body because they can be really useful tools in a lot of different settings. For example, when someone has diabetes, it means that their body can't make the protein insulin anymore to help regulate their blood sugar. So, as a treatment, they take insulin that has been produced in a lab. If we're thinking about this in the context of space, what if you have an astronaut that has diabetes or some other disease? They might need medicines to treat their disease on the ISS, and some of these medicines may be protein based. Scientists can also make certain types of proteins to be used in vaccines to help train our bodies fight against viruses and other infections. And again, you can think that it would be really useful to be able to make vaccines on the ISS to protect the astronauts if something breaks out up there. Proteins are also a very useful tool in biology research and experiments. Scientists and researchers here on Earth often make proteins in the lab in order to study them or to study other biological components. Using proteins as a research tool would be also useful on the ISS, especially in the lab, where they are currently learning more about the impact of microgravity on biology. And as a final example, there are certain types of proteins that can help perform biochemical reactions that result in biofuels. Not only would this be useful on Earth to find a sustainable fuel source that's better for the environment, but also useful out in space because we will need a lot of fuel for long distance travel, and it would be good to have a sustainable source of making fuel. So, how do proteins get made, whether it's in our bodies or in the lab? The instructions for making all these proteins are found in DNA. Our DNA contains tens of thousands of genes, with each gene encoding the instructions for making a specific protein. For example, our DNA contains a gene that encodes insulin, and your body will use this information in the insulin gene to make the insulin protein. Same thing in the lab. As long as we have the DNA that has the instructions for making all these different proteins, scientists are able to make all sorts of proteins in a lab setting. Let's go back to our example where we want to make insulin for diabetes patients. To make the insulin, scientists first take the DNA sequence that encodes insulin, or the insulin gene, and inserts this DNA into living cells. These living cells are typically bacteria, although cells from different types of organisms can be used too, and are used as a factory to produce the protein we want. Scientists grow a bunch of these cells up in a liquid culture, usually done in these huge tanks, and as these cells grow and divide, they will also take the DNA we gave them, follow the instructions encoded by the DNA, and make the insulin. We then harvest or extract insulin from the cells, and then we have the protein we can use for patients. Scientists are pretty good at doing this at a lab or at a company, but it's actually really hard to do this outside of a lab without a ton of resources. You often need to grow these cells in giant tanks like this setup shown here. And not only is this expensive, but all this equipment is really heavy and takes up a lot of space. All of this definitely would not fit on the ISS, for example. And knowing how to use all this equipment and grow the cells can also be difficult. Many astronauts don't have the scientific training to be able to make proteins like this. And finally, as you might have heard from other talks in this section, unwanted microbes, biofilms, or other contaminants are a huge source of concern on the ISS, and these sort of potential biohazards can easily arise when dealing with growing live cell cultures like this. There is an alternative to using live cells as your protein making factory: cell-free protein synthesis. How this works is that we can take those bacteria cells we normally use for protein synthesis and extract all the necessary components out, the molecular machinery involved in making proteins from the instructions found in DNA, add some supplement materials like the biochemical building blocks that make up proteins and a biological energy source, and all of these materials can go in a tube. And just to give you a sense of scale, these tubes are much smaller than those giant cell culture tanks. As you can see, many of these reactions in the tubes fit in the palm of my hand here. Everything you need to make the protein has now been combined into a single tube. Since there is nothing living in this tube, there is no need to keep anything alive, so no need for incubators, giant tanks, there is less of a biohazard concern, and all you have to do is add the DNA that encodes the protein you want, and this reaction will make it. Very easy, straightforward, and no complex protocols needed. To make these reactions even easier to work with, scientists have found a way to make these reactions more stable so they don't have to be kept super cold. This is a process called freeze drying, and we actually do this to food being sent up to the astronauts on the ISS, like this freeze-dried ice cream here, where we take whatever you're freeze drying, freeze it solid, and then put it in a special machine called a lyophilizer, and the lyophilizer will suck out all of the water in the material until all you're left with is this solid. Now, it is stable at ambient temperatures. We can do the same to these cell-free reactions. Freeze them solid and then suck out all the water until all we're left with are these little solid pellets shown here. Now, these are stable at room temperature. We can more easily ship them up to the ISS without worrying about keeping them cold. And when they get to the ISS, the astronauts won't have to worry about them staying cold until they need to use them to make proteins. With these freeze-dried cell-free reactions in tubes, now we have a portable system for making proteins. This will allow us to make proteins at the point of care. Instead of making the protein at a lab facility and shipping it where it needs to go, we could just make the protein when we need it, where we need it. You can imagine how this would be useful on Earth. For example, these freeze-dried cell-free reactions can be used to make vaccines to protect against certain infections. We've all heard the struggles of transporting the COVID vaccine and other types of vaccines to more rural or isolated communities. Instead of having these vaccines made in an industrial lab and then having to transport it, what if we were able just to transport the freeze-dried cell-free reaction, which is a lot more stable, to the point of care and make the necessary vaccines on demand at the location where it is needed? Besides vaccines, making other proteins on demand, like ones involved in medicines and therapeutics, would also be very useful. Again, using cell-free protein synthesis to do so means that we potentially don't need to ship these proteins to the place where we need them, but instead it can just, again, make them at the point of care where they're needed. We can also leverage the cell-free system and use the protein it makes as a visual signal or indicator in diagnostics to screen for pathogens or other biochemical molecules. For example, there are lots of potential contaminants in our drinking water, from metals to antibiotics. And scientists have designed a water quality monitoring assay where you can add a water sample to a cell-free reaction, and a clear color change indicates the presence of a specific contaminant. The cell-free reaction contains a biosensor that activates the transcription of a reporter gene only in the presence of the contaminant. So if the contaminant is present, the reporter gene is transcribed and then translated into an enzyme. And the resulting enzyme catalyzes the conversion of color as substrate into a yellow product, which indirectly tells us the contaminant was present in the sample being tested. All these examples revolve around the idea that cell-free systems are useful because they are portable and easy to use. Obviously, that has a lot of potential on Earth, but also has a lot of value on the ISS or other spacecraft as well. You can imagine a system like this being very useful in space, as water is a limited resource on the ISS, and being able to easily monitor water quality would be important. Another real-life example of when a system like this would have been useful was detailed in a study published in 2020 about an astronaut who had developed a blood clot on the ISS. The solution was to give the astronaut blood thinners to prevent the clot from growing, but the ISS had limited supply. Luckily, they were able to send up a resupply of the medicine before the astronaut ran out, and they were able to make a full recovery. But this raised a lot of concerns. What if the additional medication couldn't be sent up in time? What if this happens during a longer mission where we can't get a resupply from Earth? If there was some sort of cell-free system on board the ISS, the issue could have been potentially solved by using that cell-free system to produce a specific enzyme and the proteins involved that can catalyze the biochemical reactions needed to make the chemicals found in blood thinners, allowing them to make the necessary medications on board without a resupply. And that's just one example. Like I mentioned at the beginning of my talk, in order to be self-sufficient in space, it would be useful to be able to produce other medicines and vaccines, to produce protein-based tools to use in microgravity biology research, or as a biosensor or diagnostic tool to monitor the ISS environment for any contaminants or pollutants. So far, to my knowledge, no cell-free protein synthesis systems have been brought or used in space, but starting next year, that will change. Through the Genes in Space competition that is sponsored by Mini PCR Bio and Boeing, we'll be launching our own cell-free system to the ISS, a system that I worked on personally, BioBits. I developed BioBits in grad school specifically for educational uses, that is, allowing students to make their own proteins using the BioBits cell-free system without needing any fancy equipment or complicated protocols, making it more accessible to more schools, especially those that don't have as many resources for science equipment. BioBits can be used to demonstrate a variety of molecular biology concepts. For example, students are able to make fluorescent proteins and use its brightly visual output. Here it's being visualized in a blue light illuminator. And it can also be applied to a variety of engaging activities to teach about DNA and proteins, such as creating their own designs made up of many different fluorescent proteins as shown here. Our BioBits cell-free system isn't limited to hands-on classroom use, however. For our Genes in Space competition this year, we incorporated BioBits into the toolkit that students are able to use to design a DNA-based biology experiment to study space biology on the ISS. To help students and their teachers out with this, we held virtual workshops throughout the school year, sending BioBits to hundreds of students and teachers to experiment with at home, to teach them about cell-free technology, and in general, just encouraging and promoting this resource in more classrooms, especially those that don't have the bandwidth to do much hands-on activities with their students. So not only are the students able to use BioBits' cell-free system to learn about molecular biology basics in the classroom, they can take it one step further and actually propose ideas of how to use the cell-free system aboard the ISS to better understand space biology. And this is great because obviously we have a long way to go before we can make proteins at the point of care reliably. We haven't even seen if a simple cell-free reaction will work in space. But that's why it's so exciting to have BioBits as part of the Genes in Space toolkit. These ideas and proposals that the students come up with could very well be translated into the next big application for space biology. Our Genes in Space winner this year, Selin Kocalar, has a project that will test the feasibility of cell-free systems in space using BioBits to test the water quality aboard the ISS. I'm going to turn it over to Selin now and let her explain the project that will be flown and carried out on the ISS next year.
Hi, I'm Selin, and I'd like to share my Genes in Space experiment, which develops a biosensor for in-flight detection of pathogenic water contamination. But first, a bit about me. I'm a rising senior at Lee High School in the Bay Area, California, and I first heard about Genes in Space while working in a cell biology lab. There, I was mentored by Dylan Barcelos, who actually happened to be a previous Genes in Space finalist. So, with his encouragement, I submitted my proposal, worked with my Genes in Space mentor, Bess Miller, and I'm now looking forward to the launch of my experiment to the ISS next year. An essential component of safeguarding astronaut health is maintaining a pathogen-free environment in space. Therefore, monitoring bacterial communities is critical during space expeditions. We've seen pathogenic bacteria put water supply at risk in the past, and we've also seen a number of pathogenic infections in astronauts, including respiratory, skin, and even urinary tract infections. Yet, detecting and monitoring pathogenic water contamination remains a challenge. Currently, water samples are prepared in space and then flown down to Earth for analysis. Bacteria are cultured, and then their genetic material is isolated and sequenced. However, on long-term space expeditions to Mars and beyond, it's not feasible to send samples all the way back to Earth for analysis. But it's also not optimal to have to perform this complex and resource-demanding process in space, so there's a pressing need for a rapid means of in-flight detection of water contaminants, and cell-free technology may be very enabling in meeting this demand. My experiment uses BioBits cell-free systems, which Ali explained previously, to monitor water quality. BioBits is great for space applications because it can be stored for long periods of time, and at any point with just the addition of water, astronauts have instant diagnostics. In my experiment, we use BioBits to optimize a biosensor that detects the pathogen Pseudomonas aeruginosa for use in space. Here on Earth, this multi-drug-resistant pathogen commonly contaminates water sources and causes infectious diseases with mortality rates up to 80%. In space, it's demonstrated increased antibiotic resistance, biofilm growth, and virulence. Now, combine all of that with a weakened immune system caused by microgravity, and you have a major threat to crew health. Already, this bacterium has had quite a history in space. It's been isolated multiple times from water recovery systems and has caused a number of medical issues. In fact, it even caused a severe urinary tract infection in an Apollo 13 astronaut who was then mistreated in space because they could not make a diagnosis. Thus, detecting and monitoring pathogenic bacteria is critical for safe expeditions in space. To detect this pathogen, we're harnessing Quorum sensing pathways, a means of bacterial cell-to-cell communication mediated through the exchange of metabolites. At the top of the P. aeruginosa quorum sensing hierarchy is the last pathway, which promotes bacterial virulence. Here's what happens. When population density is low, the quorum sensing metabolite involved in this pathway, which is represented here in yellow, is found in low concentrations. As a result, provirulence genes are not expressed. However, as population density increases, cells exchange higher levels of this quorum sensing metabolite. Once it enters the cell, this metabolite initiates expression of provirulence genes. Now, what if we swap these provirulence genes for a fluorescent protein? Every time this quorum sensing metabolite is present, we would see fluorescence, and if it's not, then we wouldn't see any fluorescence. And this is exactly how the biosensor works. Using BioBits to express this genetic circuit, we have a system that fluoresces red when this quorum sensing metabolite is present in a water sample. Put together, we have a user-friendly, portable, inexpensive, and safe means of pathogen detection with zero sample preparation necessary. Within hours, astronauts can check if a bright red color is produced under the Genes in Space fluorescence viewer, thereby confirming whether this pathogen is contaminating their drinking water. Ultimately, through this experiment, we're not only developing a biosensor for rapid in-flight detection of pathogenic water contamination, but we're also opening avenues towards so many other applications of cell-free technology, including on-demand vaccines and wearable diagnostics. Cell-free technology has the power to redefine the future of safe space travel, And one day, this biosensor could be part of a suite of biosensors that aid in the diagnosis and prevention of in-flight medical crises.